So greetings, everyone, and good morning, good afternoon, or good evening, depending on where you are on the globe. I'm Alcidia. I'm a member of the Next Generation of Vaccinologists Committee, which is hosting today's webinar. So this is one of our monthly webinars, and I would like to welcome you to it. So as you know, or for those of you who are not familiar with it, our webinars feature two speakers, an established researcher. Today we will have Dr. Isaac Sakala and an early career researcher, Dr. Laura van Moortel. So before I introduce the speakers and let them share their insightful science with us, I would just like to share a couple of slides on who we are as part of the International Society for Vaccines and what we do. So the Next Generation of Vaccinologists, in short, Next Gen Vac Committee, is a part of the International Society for Vaccines, the ISV, which is a global, not-for-profit international organization aiming to encourage, establish, and promote the development and use of vaccines. So we would like to thank you for being here and remind you that by participating in this ISV educational activity, you are acknowledging the vital role that vaccines play in public health and in saving lives. And you're also endorsing the mission of ISV and vaccine science. So thank you again for participating in our activities. So today's webinar, as I mentioned, will feature Dr. Isaac Sakala and Dr. Laura van Moortel, and our webinar will be based on the theme of tuberculosis. And as I mentioned, these are monthly webinars, so you can already save the date for the next one on the 5th of March, where we will have exceptionally two early career researchers, Ines Corrives and Dr. Juan Garcia Bernal. More information will come on our official platforms, so make sure to follow us there to get all of the information, including the Zoom link. The activities of the ISV would not be possible without support. So this is why the industry committee was established, to ensure sustainability of our society, as of course funds are needed to support our mission and our programs. So we would like to thank our annual supporters and board members for their commitment and or their financial support. So if you would like to join and support the activities of the International Society for Vaccines, or just to get more information, you can contact Ted Gibson at the email address on the screen. So as I mentioned, we are featured in all the different social media platforms for which you can find the QR codes here. So make sure to follow us there and stay tuned for our latest news. You also have the possibility of becoming a member of the ISV, which gives you a plethora of benefits, including access to travel awards, mentoring, professional development, discount on the ISV annual congress, journal access, and global networking opportunities. And we have membership rates depending on where you are from and the degree that you are at your career. So these are all very interesting. So if you are not yet a member, I would highly encourage you to join the ISV. As I mentioned, each year we have an annual congress, so save the date for this year's one, which will occur mid-October in Antwerp, in Belgium. And coming back to today's webinar, so we will have one talk by Dr. Isaac Sakala, our expert speaker today, and one by Dr. Laura van Moortel. In between the two talks, we'll take one or two burning questions, which you can write down in the chat as participants. And at the very end of the two talks, we will have a short Q&A session with questions that can be for either or both speakers. So feel free to write them all throughout the webinar. So without further ado, I will introduce our expert speaker, Dr. Isaac Sakala. So he is a senior research scientist at Centenary Institute in Australia within the Tuberculosis Research Program and Advancing Vaccine Adjuvant Research for Tuberculosis NIH contract. In addition, he holds an adjunct associate professor position at the Australian Respiratory and Sleep Medicine Institute. He earned his PhD in immunology from the Australian National University in 2008. After that, he moved to the US, where he undertook postdoctoral research fellowships at St. Louis University and Washington University from 2009 to 2015. During that period, he focused on identifying gamma delta T cell specific antigens in mycobacteria and elucidating the roles of host cells and mucosal associated invariant T cells in protective immunity against mycobacterial infections. He went back to Australia in July 2015, and since then he contributed his expertise to the research efforts at Vaccine Flinders Medical Center, and here he focused on studying novel vaccine adjuvants aimed at optimizing vaccine priming in neonates and young infants. His research also involved characterization of these adjuvants' mechanism of action in inducing robust and protective vaccine immunity. So, Dr. Sakala, the floor is yours.
Thank you, Alcidia. Let me share my slides. I think you are still sharing? I'm not sure.
Maybe you can try sharing again.
I can't see my slides. I think you are still sharing. I'm not sure if you exited your presentation.
Yes, I did.
You did, okay. Yeah, okay. Got around it. All right. First of all, I'd like to say thank you to the Next Generation of Vaccinologists Committee for inviting me to share our data on the vaccine development here at Centenary Institute. TB for the last 200 years has been one of the leading causes of death from a single infectious diseases. And one in third of the world population is infected with TB. And the WHO reported in last year that about 10.7 million new cases of TB were reported, and about 1.3 million deaths that resulted from TB. And the burden of TB really varies. That is in terms of the new TB cases varies from one country to the other. For example, in other countries like the Americas and Europe and Australia, you have an incident rate per 100,000 population per year of less than 10, while in other countries you have more than 500 cases per 100,000 population per year. Next. And there are about 30 countries throughout the world that have got an incident rate of more than 100,000. And out of these, 8 of those contribute about 65 or 67% of the total TB burden in the world. So what are the challenges of TB control? We do have a vaccine, which is BCG, that was developed in 1906. And this vaccine has been administered for more than 100 years. It's estimated that more than 3 billion doses have been administered. BCG itself has got limited protection in adults and adolescents, but has been very good at protecting against early childhood TB and most severe complications of TB. You can see here in the study that was carried out, there's a lot of variation of protective efficacy in each and every country across the world. The second problem is that there is development of multi-drug resistance. So this is resistant to the first-line treatment drugs, isoniazid and rifampicin, and also extensively drug resistant, that is resistant to the first line of treatment and the second line of treatment. So in 2024, it was estimated that about 300,000 cases of MTB are among the world's 10.7 million prevalent cases. And you can see here that most of these cases are in those places where there is high burden of TB. So to reduce the global TB burden, really we need new generation TB vaccines or vaccination strategies. So these vaccines should be able to induce persistent immunological memory to protect beyond the first 10 years of life. Because when at birth BCG is given, it does help protect against children, but as they grow, the immunity wanes off, and by the time they are adolescent, which is the age that really is responsible for much of transmission of MTB, there is no protection against MTB infection. So in 2019, there was a paper that reported that that M72 fusion protein that was formulated with a saponin-containing adjuvant provided near 50% of protection against MTB-exposed adults, and that was sustained for over 3 years. This marked really a major milestone in TB vaccine development for 2 reasons, because it showed that we can generate vaccine-induced immunity against TB. Secondly, it also showed that there is need for development of TB targeting adjuvants. We still have a challenge that is it possible that we can make a vaccine or create a vaccine that can induce protection more than 50%, and that would be very helpful and have an impact on the TB pandemic. As of January this year, there are about 9 candidates of TB that are in active clinical trials, and they include these vaccine types: live attenuated, inactivated, viral vector, protein subunits, and the messenger RNA vaccines. And they are aimed at preventing infection and also preventing disease development, and also preventing reactivation of TB, and also that they can be used as therapeutic vaccines along with the standard treatment of TB. Here at Centenary Institute, we are involved in a number of TB related or TB vaccine development programs. We have a group at the University of Sydney that is focusing on the development of RNA vaccines for TB, and also viral vector vaccines that can be delivered to the lung, which is the site of infection. And we're also involved in just identifying the correlate of protection in the lung. But we're also involved with identifying, as I said, adjuvants that can help drive the desired TB immunity in vaccines. And to that effect, we have funding that was given to Professor Britton and the ISO and the Trickers here at Centenary Institute and the University of Sydney that is allowing us to test or evaluate 10 different adjuvants that target different innate immune receptors. And these are highlighted in red, and the receptors that they engage in order to induce an appropriate immune response. So we hypothesize that the ability of these adjuvants to engage specific immune pathways will be key to unlocking, effective and durable TB vaccines. So this is the AVRT program that the NIH contract that was awarded to Professor Britton, ISO, and Trickers. We are performing very systematically side-by-side comparison of TB proteins mixed with adjuvants to identify which adjuvant is capable of mediating protection. Most of this work is done here at Centenary. We're looking at the safety, immune responses in terms of the early immune responses and the adaptive immune responses, and also transcriptomics, and also looking at the ability of those different formulations to induce an immune response that is protective against LOS or MTB challenge. We have a site also in the USA, in Oregon, where they are looking at the effect of these adjuvants on human cells, like dendritic cells, which are taken from humans, and looking at how they influence the activation of different innate cells. And after all this is done, two prominent or promising adjuvants or formulations will be tested in non-human primates at the Today National Biomedical Research Center. So this is the whole pipeline from preclinical, from mouse work and to non-human primate, and hopefully it can end up in the human clinical trials. So we have 10 adjuvants, as I said, and we have also 3 candidate multi-stage TB fusion proteins. The selection of these 3 fusion proteins that we are targeting the early and chronic stage of infection, so that they can be effective prophylactic and also therapeutic vaccines. So they are also able to induce broad immune responses that we hope can protect against TB. Just to remind ourselves of the immunity to MTB, innate cells, different myeloid cells are important in just controlling MTB at the site of entry in the lung, and also just involved in the activation of adaptive immune responses. And in terms of the adaptive immune response, we have the CD4 T cell that is very established to be very critical in protecting against the MTB. So these CD4 T cells can differentiate into subsets that are very critical, like Th1 that produce these cytokines here, and Th2 that is also involved in B cell activation, and Th17 that produce these cytokines, which are very important. B cells and these CD8 cells are increasingly being looked at as also important in the control of MTB. So just to show you some data, we are looking at the early innate and adaptive T cell responses to post-vaccination. In this experiment here, we have a single dose and we get organs at different time points just to look at the phenotype of those cells and also the functions like in cytokine production at all these time points. And at day 7, we also look at the T cell responses in terms of tetramer positive cells, as well as ability to produce the cytokines. So this is a heat map showing recruitment of myeloid cells to the draining lymph node after a single dose. And you can appreciate here that there is a variability in terms of recruitment of various innate cells to the draining lymph node. You can see ADVAX CPG predominantly induced these macrophages here, and then T-vant really induced a lot of innate cells. But you can see also here that this hydroxyquin and kappa-1 were really silent in terms of inducing myeloid T cells recruitment to the draining lymph node. This data is for the increase with relation to the saline control and the vaccinated groups. And we also looked at the early T cell responses, that is after a single dose. These are just the flow representatives showing CD44 marker, as well as the MH class 2 restricted and antigen 85B tetramer positive. You can see that there was no difference. They were very comparable among all these 3 initial adjuvants that we looked at. This shows that these 3 adjuvants are able to prime the immune response. And we also looked at the second set, which is ALF-Q, Mimvax and T-vant adjuvants, and you can see also that there was no difference at day 7 in the draining lymph node in terms of inducing early T cell responses. We've also looked in the lung and the spleen, and we can detect them at day 7. Moving on from early single dose, now we also looked at the ability of these adjuvants to induce memory T cells following 3 doses. And this is just a schematic diagram to show the scheduling of vaccination. We wait for 30 days and pull out these organs and look for immune responses using tetramer staining and intracellular cytokine staining. This data shows the average percentage of tetramer positive cells that were measured in the spleen and the lung after the 3 vaccinations, Sysvac2, H56, and the PM. And you can appreciate here that at the day 30, ADVAX CPG in red bars induced the highest antigen-specific T cells, both in the spleen and in the lung. And it was like that for both in the Sysvac2 and H56. And also at 120 days, there was persistence of these antigen-specific CD4 T cells only in the Sysvac immunization. But if you look at AHQ and kappa-1, there were similar responses at day 30 and only persisted in the H56 in the kappa-1 vaccination. If you look at the PM immunization, we are similar, AHQ as well as CAFA3, CAFA1, but only CAFA1 had persistent T cell responses. And this is the functions now of those CD4 T cells using intracellular cytokine staining at day 30 as well as long-term to measure durability of those memory cells that we were able to detect at day 30. And this is showing a scatter graph showing interferon gamma production, TNF, interleukin-2, and interleukin-17 at day 30 and day 120 after the last dose. And you can see that the Advax CpG was very good at inducing interferon gamma, TNF alpha, but CAFA1 was better at inducing interleukin-17. And the hierarchy was maintained even at the 120. You can see Advax CpG had higher interferon gamma, while CAFA1 had higher interleukin-17 producing CD4 T cells. So different adjuvants induce different patterns of cytokine producing CD4 T cells at day 30 with the hierarchy maintained at 120. And the pattern is similar in the lungs. You can see that interferon gamma producing T cells are mostly induced by Advax CpG, and while interleukin-17 producing CD4 T cells at 120 days was only sustained by CAFA1 adjuvant. So that's the first set of the adjuvants that we tested. And this is now a similar protocol. We tested ALF-Q, which is a toll-like receptor for targeting adjuvant, and the MimVax and TVANT, a similar protocol as I explained. And I'm showing you tetramer positive T cells after Factor vaccination and H56 vaccination. You can see ALF-Q has the highest antigen-specific CD4 T cells at day 30, both in Factor and in H56 vaccination. MimVax had the small but significant antigen-specific CD4 T cells in H56 immunization. TVANT was the least in inducing the antigen-specific T cells at this time point. We have not yet done with the 120 in the CISFAC2, in the H56, but in the lung now, I'm showing you interferon, I'm showing you cytokine production after protein restimulation at day 30, both in the spleen and in the lung. And you can see that ALF-Q was really the best adjuvant here at inducing Th1 cytokines as well as the Th17 cytokine in the spleen, and it was similar pattern as well in the lung. And this is only in the CISFAC2 immunization. We are yet to harvest the H56 data. So I'm showing only CISFAC2 immunization in the spleen and the lung at day 120. And you can see that the memory immune response we see at day 30 persists to day 120 in the ALF-Q adjuvant groups, which is good because it shows that it's long term. And so I'm going to show you some data whether this immune response is able to protect against the aerosol challenge. So to summarize on T cell immunogenicity, we can see that there are three adjuvants that we've seen they can induce the early T cell proliferation. I haven't shown you this data, but they can also recruit to the T cell to the lung. They can polarize Th1, but not Th17, but they can also have memory T cells at day 30 and also long-term memory T cell at day 120 for Advax CpG, CAFA1, as well as ALF-Q. Now this is protection data, and so we after 3 doses of immunization via the IM route, we wait for 30 days, and then you do LSO challenge and harvest at day 30 after LSO challenge to look at CFUs. And this data is showing a mean log 10 CFUs in the lung at time T30 for Sysvac immunization, as well as the H56 immunization for these adjuvants, Advac CPG, CAFA1, and AHQ. And it's summarized here that you can see that the Advac CPG provides protection in both the Sysvac2 as well as H56 vaccination, and as well as CAFA1 does confer protection in Sysvac2 as well as H56 immunization. The list was AHQ, which is a toll-like receptor 7/8 targeting adjuvant, while Advac CPG targets toll-like receptor 9 as well as DC sign. CAFA1 is a MINCO adjuvant. And this is the other second set of adjuvants. I showed you that ALFQ was the best at inducing memory T cells, and here you can see that it does protect against the MTB both in the lung and the spleen. MIMVAX does protect in the spleen. However, it was non-specific, because if you look at MIMVAX, there was no difference between the control, despite that there was that is in the spleen. There was no difference between the control or adjuvant by itself and the adjuvant with the antigen. And so this is non-specific, and also for TVANT non-specific. But this one is TB specific. This data just shows you the log reduction as well. So we also have a scheduling whereby after three doses, we wait for 120 days to challenge those mice, and then harvest the organs for CFUs as well as immune responses. And so here we are measuring memory protection after waiting for 120 days. Those T cells able to protect against the aerosol challenge. And you can see here that ALFQ, both in the lung and the spleen, is able to induce protection after waiting for 120 days to challenge those mice. So in summary, I've shown you that just a single dose of Sysvac2 with these adjuvants can induce antigen-specific T cells in the draining nerve by day 7. Advac CPG enhances antigen 5B tetramer positive cells in the 2 vaccinations, and is sustained to 120. For the other set of adjuvant, we've seen that the ALFQ is the best performing adjuvant. And so that's very promising for development of the next generation TB vaccines. So just to summarize that, out of these 10 adjuvants, we are leaning towards selecting ALFQ as well as which targets T-like receptor 4, and Advac CPG, which targets DC sign and Toll-like receptor 9. So these vaccines will be tested in monkeys at the Turin National Biomedical Research Center. So that was in adult mice. We are also developing a model in newborn mice, so that we can test these vaccines in the context of an early immune early life situation. So we've selected just 4 adjuvants that we are going to evaluate in newborn mice. And this is just an overview of a newborn TB vaccine project that we want to do. And we'll have newborns at 1 day old or 7 days old, and looking at just various immune responses, the T cells, the B cells, and long-lived trained immune responses. just as a proof of concept, we have shown that advax CPG is immunogenic in newborn mice. These were 7 days old mice that we have given 1 week apart, and they waited for 30 days, and they did intracellular cytokine staining. The data that I'm showing you here in the draining lymph node, the spleen, and the lung, we are able to detect Th1 cytokine responses. And these are just co-expression of interleukin-2, interferon gamma, and TNF in the same paths. So in the newborn mouse TB vaccination and challenge model, we have established that Sysvac2 and advax CPG vaccine is immunogenic in newborn mice. We are going to go ahead and determine if newborn 20 microgram dose that we're going to use is protective, and then characterize the immune responses with various adjuvants. And so we are also in the middle of looking for funds just to support this neonatal project. So I'll end here, and thank you very much for listening.
Thank you very much, Dr. Sakala, for this very insightful presentation on vaccine adjuvant research for tuberculosis. Very interesting. For the sake of time, we'll go directly to our second speaker's presentation and answer the questions at the very end. And for the chat, I highly encourage you to ask questions, which will be answered by the speakers in a couple of minutes. So it is now my pleasure to introduce Dr. Laura van Moortel. She's a postdoctoral fellow in the lab of Professor Impens at VIB and Ghent University in Belgium. After obtaining her master's degree in biomedical sciences, she did her PhD in the labs of Professors De Buscher and Gevert, also at VIB, and there she focused on the characterization of potential better ligands for the glucocorticoid receptor using cell and proteomics-based approaches. After her PhD, she joined the team of Professor Impens to work on the EU-funded Vaccinologists project, where she used immunopeptidomics to identify candidate antigens for the development of novel mRNA vaccine formulations against Mycobacterium tuberculosis, Acinetobacter baumannii, and Mycobacterium ulceronans. So, Laura, it is my pleasure to give you the floor.
Thank you very much, Alcidia, for the kind introduction. Okay, can you see my slides?
Yes.
Okay, perfect. So yeah, thank you very much to the organizers for the opportunity to present the work that I've been involved in for the past 2 years, which is the development of novel mRNA vaccine candidates for MTB using immunopeptidomics. So I think I can more or less skip the first slide since this was already very nicely introduced by Isaac. So there is still a very big need for novel vaccines against MTB, but one of the difficulties in that and also for the development of novel vaccines against other bacteria or complex pathogens is that it's quite difficult to determine which proteins could be good candidate antigens for a vaccine. And so this is where immunopeptidomics comes into the picture. And so this is actually the expertise of my host lab, so of the team of Professor Francis Impens. And so immunopeptidomics is a mass spectrometry-based technology that allows you to identify human or bacterial peptides that are presented on major histocompatibility complexes. And just to introduce this very briefly, just to make sure we're all on the same page. So there are 2 different classes of major histocompatibility complexes, or also known as HLAs. So you have MHC or HLA class 1 that is expressed by all mammalian cell types or by all nucleated cell types. And these MHC class 1 molecules will present peptides that come from intracellular proteins, so either from the host or from pathogens that were processed by the proteasome and that are then loaded on MHC class 1. And so these are recognized by CD8 positive T cells that then assist in cellular adaptive immune responses. And then secondly, you also have MHC class 2 that is only expressed by professional antigen-presenting cells. And on these molecules, peptides will be loaded that come from extracellular proteins from the host or from pathogens, and that were degraded by the endolysosomal system. And so MHC class 2 is recognized by CD4 positive T cells, and this then helps assist in humoral immune responses. And so to go back to immunopeptidomics, what we do in the lab, so we infect usually human cell lines with our pathogens of interest, and then we lyse them using a mild lysis buffer. And next, we perform an immunoprecipitation step for the MHC class 1 or class 2 complexes using specific antibodies. And then we elute these complexes under mild acidic conditions. And then we do a cleanup step to remove all potential salts or other contaminants before injecting these peptides on the mass spectrometer for identification. And so the proof of concept for the use or the beneficial strategy of immunopeptidomics for bacterial antigen discovery was done by our former colleague, Rupert Meyer, who did his postdoc in the group of Francis working on Listeria monocytogenes, which is an intracellular foodborne pathogen for which it is known that the protective immunity solely depends on MHC class 1 and CD8 positive T cells. And so what Rupert did was infecting human cells with Listeria and then subsequently subjecting these samples to immunopeptidomics. And then so he looked at all the bacterial peptides and proteins that were identified in this screen. So on the x-axis, you can see all the bacterial proteins. And then on the y-axis, you can see the number of immunopeptides that was identified per Listeria protein. And then so our hypothesis was that the Listeria proteins for which you find the most immunopeptides, that these are the proteins that are most seen by the host immune system during a natural infection, so that these likely could be the best candidate antigens for a vaccine. So then they evaluated this hypothesis by generating mRNA liquid nanoparticle vaccine formulations encoding each of these top antigens. And then they did prime boost vaccinations in mice, followed by a challenge with Listeria. And then they looked at the number of colony-forming units in the spleen and the liver as a measure for the bacterial loads. And then what they could nicely see is that Lmon 0149, which is the Listeria protein for which we found the highest number of immunopeptides, that this was also the most protective against infection with Listeria in mice, leading to more than 95% reduction of the CFU. And this result could also be nicely recapitulated over multiple biological repetitions. So this was then the proof of concept and kind of the starting point for Baccerna. So this is the project that I have been working on for the past 2 years, and that is funded by Horizon Europe. So it's coordinated by VIB, so by Professor Francis Impes and by Professor Lennart Martens at VIB. And it consists of 10 different institutes across Europe that are involved. And the ones that I have highlighted here were involved that I'm in the work that I'm presenting in the next few slides. So the goal of Baccerna is to develop novel mRNA vaccine candidates, not only against MTB, but also against Mycobacterium ulcerans and against Acinetobacter baumannii. But for the sake of time, I'm only going to discuss our work on MTB. And the overall strategy was very similar as the work that was done on Listeria. So for the antigen discovery, we are again using immunopeptidomics, now with a more high throughput sample preparation format, where we can now do subsequent IPs for MHC class 1 and class 2 on the same sample in a 96 well plate format on a positive pressure and liquid handling system. This work is currently under revision, so hopefully it will be published soon. And then for the data analysis, we also have an integrated pipeline that was developed by my colleague Patrick, and that has also been published in the meantime. So using then this pipeline for MTB, this was done in collaboration with the Pasteur Institute, with a group of Caroline Mongel, where THP1 and UNIT37 macrophages were infected with MTB. So these are both human macrophage cell lines. They were infected for 24 or 72 hours, and then subsequently we prepared the samples for immunopeptidomics using the same approach as I previously explained. And then looking at the data, we always first do a number of quality controls to make sure that our immunopeptidomics data is of good quality. So across all the screens in these two different cell lines, we found more than 30,000 MHC class 1 peptides, of which most come from human proteins, but we also found MTB peptides in there. And what we expect to see for MHC class 1 data is that the vast majority of the peptides that we identify, that these should be 8 or 9 amino acids in length, because this is the optimal length for a peptide to bind to an MHC class 1 molecule. And what we could also see is when we perform MHC binding predictions using NetMHC-PAN, is that the majority of these immunopeptides are predicted to bind at least one of the MHC alleles that is expressed by UNIT37 or THP1. This you can also nicely see here in these peptide sequence logos. So on the right are the theoretical logos that we expect for the HLA class 1 alleles that are expressed in THP1 and UNIT37. So these were also retrieved from NetMHC-PAN. And then on the left, we did unsupervised clustering on all peptides between 8 and 12 amino acids long. And then you could see that the different clusters that were formed, that the peptide sequence logos of each of these clusters very nicely matched the theoretical logos that we expect, again, confirming that we had immunopeptidomics data of very good quality. So then the next step was, of course, to look into the MTB peptides and proteins that we identified, because that's, of course, what we were most interested in. So we took our own data, our own experimentally generated data, and then we also performed a reanalysis of publicly available immunopeptidomics data from human primary macrophages that were infected with MTB. And then we looked at all the MTB peptides that were found in all of these data sets, and then did a number of filtering steps to make sure that all the peptides that we ended up with were confidently identified and not false identifications. So this was 199 MTB peptides from 163 proteins. And then we narrowed that a little bit further down. So we again zoomed in on the MTB proteins for which we found the highest number of peptides, or MTB proteins for which we found one peptide that was a predicted MHC binder. And then from this list, we again selected 15 candidate antigens that I unfortunately cannot disclose yet, but hopefully we will be able to do that in a few months. But so these 15 candidate antigens were then prioritized again based on how many peptides that we found, also on the MHC binding predictions. And then also very importantly is we checked that all of these candidate antigens are highly conserved across different MTB strains. And what we also checked is that these antigens are poorly conserved again in common host microbiome species, because you want to avoid developing, of course, a vaccine that induces immune responses against harmless pathogens. So then we ended up with these 15 candidate antigens from our immunopeptidomics screen. So then, of course, the next step was to evaluate whether these could indeed be good candidate antigens for a vaccine. So we again generated mRNA liquid nanoparticle formulations in collaboration with the VUB and Ghent University. And for the sake of time, and also because these MTB challenge experiments had to happen in a BSL-3 facility, we did not have, we could not test all these antigens individually. So we immediately paired them up. So we in total had 8 formulations to test. And so the challenge studies were again done in the Pasteur Institute, where the mice were vaccinated twice with 2 weeks in between, a prime boost vaccination with the mRNA LNP formulations. And then at 6 weeks, the mice were subjected to an aerosol MTB challenge. And 4 weeks later, the mice were sacrificed. And we counted again the number of colony forming units in the lung and the spleen. And so these are the results from our first challenge studies. So the results in the lung and in the spleen. We always compared our candidate vaccines against unvaccinated controls and against BCG, since this is currently still the only licensed MTB vaccine on the market. And so what you could see is that in this first challenge study, that the effects of our mRNA formulations were still relatively mild, although we did already have one formulation, G, that already had significant protection in the lung. But what we also know in particular for mRNA vaccines, is that if we combine multiple antigens into one multivalent vaccine, that this can really boost the protective efficacy. So that's exactly what we did. So we took formulation G, H, and C because these had the most promising effects in the first challenge experiment. And we combined the 5 antigens in these first formulations into one pentavalent mRNA vaccine that we then again tested and compared to BCG. And we were very happy to see that now, in particular in the lung, that the protective efficacy of our pentavalent vaccine is very closely matching the effect of BCG. So we are very happy with that. And then what we also did is, since most countries still perform routine vaccinations with the BCG vaccine, we also tested what the effect would be of our vaccine candidates when administered as a booster for BCG. So this is what you can see here on the right. And so what we could tell from this last experiment is that when you administer our pentavalent mRNA vaccine as a booster for BCG, that it leads to a protective efficacy that is better than BCG or the mRNA vaccine alone. So we are very happy with the results that we have been able to get so far. So all of this work more or less happened in a time frame of 2 years, but Baxerna is still running for a few more years. So the next steps that we are planning is immunogenicity studies in non-human primates. We're also still gathering actually immunogenicity data from the mice. We're also going to do toxicity studies in rats. And in the meantime, other partners in the consortium are already setting up the GMP production of the vaccine candidates, because the goal is that we would be able to start a phase 1 clinical trial at the end of 2027. So yeah, we're very happy with the data that we have gathered so far. And so with that, I would like to thank all the people that were involved in this work, because this has really been a huge team effort where everyone brought their own particular expertise to the table, which also allowed us to advance this work quite quickly. I would also in particular like to thank my supervisor, Professor Francis Impens. And then I would also like to briefly advertise a little bit for our upcoming Baxerna conference, which will take place in Belgium at the end of March. So the official abstract deadline has already passed, but we will still accept abstracts for posters, and the early bird registration deadline is February 11th. And then I would like to thank you very much for your attention, and I'm happy to take any questions.
Thank you, Laura, for this brilliant presentation. So let's jump straight to the questions, as we have only a couple of minutes left. So there was a first question for Isaac from the chat, and it was, at what stages are the adjuvant, if you can disclose? And if maybe combined to that question, if developing a new vaccine, wouldn't it be less risky to use maybe established ones, or is it that the pathway is maybe not suitable, for example, for aluminium salts?
The adjuvant, the adjuvants that we're using, some of them are already in clinical use. Some are already being developed as preclinical adjuvants. And that's why it's important that we test them in reactogenicity and causing problem in mice. Then the chance of it progressing further, it's diminished. That sort of stuff, yeah.
Okay, thank you. And to continue on that, were the adjuvant doses pre-titrated, as in, did you know which doses were needed for each adjuvant, and could they be disclosed, or are they in maybe?
Yes, yes, we did, yes. So, and they have tested those prior to them giving to us, and so we know the optimal dose of the adjuvant to use. If we are not sure, we do go back to them, and then they suggest what range or what dose that we can use. It happened, for example, with the Tivant. We had used a higher dose, like 10 microgram per mouse, based on their publications. But we found that it was very reactogenous. The developers who suggested a lower dose of the adjuvant.
Okay, thank you. I have a question for Laura. So you presented combinations of adjuvants as you established many different candidates which could not necessarily be tested each on their own. Have you also considered combinations with known adjuvants, or is it really only novel adjuvants? And would that be interesting?
Quite a number of the antigens that we have selected and that we have evaluated were known and are also being tested in other MTB vaccines that are in the pipeline. But we also had a few antigens that were more novel. So we're actually very happy with this because if we would find none of the very well-known TB antigens, it might also lead us to question our own data. But of course, it's also very nice that we did find some novel antigens that were not previously described yet as potential vaccine candidates.
Thank you. And when discovering those antigens, did you need afterwards to do specific modifications to the mRNA to fit those adjuvants? And how does that, if you can disclose, compare to what has been done to other commercially available mRNAs?
So you mean in particular then the adaptations to the mRNA? So yeah, we have codon optimized it for expression in mammalian cell systems. We also checked whether there were any secretion signals in there that need to be removed because these would not be recognized by the mammalian cell machinery anyway.